Good morning. How's everyone doing today? Thanks for coming to the video broadcast this morning. How's everybody doing? Good morning, Eli. Good morning, Cole. Good morning, Melody. If anybody else wants to check in, please feel free to do so on the chat bar. How's my sound coming through? If you could give me a quick sound check, make sure that you can hear what I'm talking about this morning. Okay, so today is the second lecture of the biopsychology chapter. Remember, we talked about each and every. Uh, we suggested that there are 52 areas of specialization in psychology, and I suggested to you that each and every chapter is going to cover one of these areas of specialization, one of these subfields. And today we're talking about biological psychology. And we're going to be revisiting biological psychology throughout the semester because the anatomy and physiology of the brain, in fact, is the foundation of all of these behaviors that we are studying. So we're going to be revisiting this over and over. I hope everybody comes to my webinar tonight at 7 p.m. The discussion board or the collaboration activity is due tonight at 11:59. You can either do the discussion board or you can come to the webinar if you want. Yes, Nicole, I just uploaded PowerPoint 2A about 10 minutes before class, and PowerPoint 2B is also going to be represented today in this lecture because I just uploaded the uh, the new version of that this morning. Good question. Thanks for asking. Hello, Jade. Good to see you. Good to see you, Ashley. Thanks, Lilibeth. And Sarah, great to see you as well. Let's go ahead and get started in the lecture. And I want to start by asking you a question. I'd like to introduce you to my 9 friends here. You see them numbered 1, 2, 3, 4, 5, 6, 7, 8, 9. How easily do you think it would be for you to tell them apart and learn their names? Would it be easy or do you think it would be difficult? What do you think, folks? I'd be curious. So that's Fred is number 1, Eileen is number 2. I've got Nathan is number 3. Uh, that's Aliyah is number 4. Sheldon is number 5. Sam is number 6. It would be hard, wouldn't it? It would be absolutely difficult, right? Why would it be difficult, Cole? Do you have any reasons? What might make that so difficult? Some might seem easy. So Nicole says that she might be able to identify them by some one of their scratches. So something unique or distinctive about that person. Right. Absolutely. Well, I tell you what, how about these friends? These are some of my other friends. Do you think you would learn to recognize them by their faces pretty easily? How easy would this be? Would you be able to tell? So we've got Shaylin is number 1. That's Edward. That's Nathan right there. That's Eileen. Then I have Ted, Jeremy, Sarah, John, and Abigail. Would that be a little bit easier for you? Absolutely right. You know what? It turns out that human beings have a special place in their brain that is responsible for processing facial memory. I'm sure all of you have had the experience of running into somebody you met before and you've forgotten their name but not their face. That's not because your memory is bad. That's because your memory is so good. Your memory for faces is absolutely exquisite because faces and facial recognition is very important part of your survival. So we've actually evolved an area of our brain that recognizes faces. It's called the fusiform gyrus, and it's located towards the bottom and inside of the temporal lobe. We're going to talk about it later today and it is very important for facial recognition. However, there are people who are born with fusiform gyruses that do not work properly, and these people suffer a disorder known as prosopagnosia or face blindness. And this is a disorder where people are not able to recognize other people's faces, even faces of people that they've known for years and years, even their parents' face, even their own face they can't recognize. Because for them, faces look like a pile of rocks, if you will. And the only time they can tell one person apart is if they have super long hair, or if they have a super big afro, or maybe if they're super tall. So, in a sense, the only thing they could do is what Nicole said. They could only see the big scratches on the rocks. And so, we actually have an area in our brain that's responsible, for doing that. And that's called the fusiform gyrus. And that's sort of going to be the focus of today's lecture. All right, now, what we're going to talk about today is we're going to talk about two brief principles of the human nervous system organization: modularity and connectivity. Okay, and they're going to be the organizing principles of the brain. And then we're actually going to go through and I'm going to point out some of the areas in the brain that we have identified the functions of. And I'm also going to suggest to you where we're going to run into them later on in this class. We're going to talk about the structures in the very bottom of your brain, the hindbrain. We're going to talk about structures that are up in the top of your brain, but they're tucked underneath your cerebral cortex, your forebrain structures. And then we're going to talk about that thing called the cerebral uh, cortex, which is what most people think of when they think of a brain. Okay, so let's go ahead and start. Now, in order to know anything about the biology and the anatomy of the brain, we have to be able to study the brain with some sort of visual or measuring technique. And the visualization and measurement of the brain has really improved in the second half of the 20th century. And so now we can actually look inside and see what live brains are doing. We can see what kind of activity they are doing, and we can see more specifically where in the brain this activity is occurring. Now, there are four basic ways of learning about the brain, and I'm going to talk about them briefly. So the first is the oldest, and this is what Paul Broca used when he discovered Broca's area a couple of hundred years ago. This is called the lesioning study. Sometimes people accidentally have brain damage and they lose a functional ability. And so a researcher can do a case study, if you remember that from last week, a case study, a specific in-depth study of a unique individual whose situation can't be recreated. A lesioning study provides an opportunity for a case study to be done. So, what you do is you look at what part of the brain is damaged and you correlate that to the behavioral deficit that the person has, and voila, you can begin to build a model of how the brain functions. Examples of this are Paul Broca in TAN, which we talked about last week, language production, or maybe I talked about it in Tuesday night webinar. That's where I talked about it. And then also, if you look in your textbook, they'll tell you the story of Phineas Gage, the railroad worker whose frontal lobe was destroyed and changed his personality. The second technique that we can use is called the EEG. It's where they place the electrodes all over your brain, and then they measure the electrical activity of these different parts of your brain. This was invented in the 1930s by a Nazi scientist named Hans Berger. And what this does is this measures the amount of electricity that's being produced by different parts of your brain at any given moment. And if you look at the far left-hand corner, you'll see an EEG readout. Now, EEGs are very important in sleep studies. When we get to chapter 4, we're going to talk, chapter 3, we're going to talk about sleep studies, and I'll talk about the electrical activity that occurs in the brain while we sleep. Turns out your brain does not go to sleep when you are. It is still awake, maybe even more awake than it is when you are not asleep. And you know what? We can also use the brain. We can use the EEG to measure these things called event related potentials. We can tell when people are surprised by novel objects because their brain activity spikes, showing that their brain has been surprised. So we can use event related potentials. Um, a third technique is right underneath me. It's called transcranial magnetic stimulation. Now, I told you that uh, we can't damage people's brain and study what behavioral deficits they lose. You can't do that as an experiment. You have to do that as a case study and a lesioning study. However, the use of TMS allows the researcher to safely disable parts of the brain for just a brief second. And then the researcher can measure what behavior changes when they disable that part of the brain. And so the TMS is a way to do sort of an experimental and temporary experimental lesion. And we can see what people can't do when parts of their brain have been deactivated. And then finally, at the very, very bottom of the screen, what you see is an fMRI, a functional MRI. And what they do in the fMRI is they inject you with a radioactive dye that tends to hook up with oxygen molecules. And then they put you in an MRI machine and they'll ask you to think about different things or to do different mental calculations. And what they do is they watch where in your brain the oxygen piles up because we know that cells use oxygen when they're working really hard. And so the fMRI can sort of tell us from moment to moment which part inside your brain is working the hardest. And if you look at the picture at the bottom of the screen, you'll see that the areas that are yellow and red are the most active at that given moment. So we can sort of see what parts of a person's brain are active when they are engaging in a thinking task. I see that we've had several people walk into class during the lecture and say hello, and I want to stop for a moment and tell each and every one of you hello, good morning, and thanks for coming today. We're talking about how to measure the different areas of the brain. Okay, now, two things I want you to know about the brain that can explain how the brain functions. The first is brain modularity. Now, there was a time 200 years ago when people wondered whether or not the brain was one computer, one processing machine. They didn't have computers back then, one processing machine or multiple processing machines. It turns out that your brain is multiple processing machines. Each of these machines or networks or areas, if you will, processes one simple task. And each one of these computers works together to create complex behaviors. So, for example, your vision is processed in pieces by different areas of your brain. One area of your brain looks at the lines that make up Dr. Roddenberry right now. Another part of your brain is measuring where I move at any given time. Another part of your brain can tell you where I am in your visual screen. Another part of your brain is simply there to record the colors that are in the image you are looking at. And then all of those areas report to a fifth area that puts it all together and figures out what you're looking at. So your brain is a series of modules that each engage in a specific simple ability, and then those abilities are integrated together. If you look at this picture right underneath me, you are looking at what's known as a Brodmann map. This was created by a fellow named Corbinian Brodmann almost 100 years ago, and he went through brains and he could tell that there were different areas in the brain based upon how the cells in that area looked, how they were shaped, how they were organized, and he found that we had almost 52 different areas on our cerebral cortex. And you can look at all of these colors to sort of see the different Brodmann areas. And these Brodmann areas control all aspects of our conscious functioning, not just our personality. Right. Now, we know what almost all of these behaviors do, but we're still learning about how these different modules work together. Okay. Now, here's the deal. If you look at this area on the brain, what you're going to see in the brain is these modules are created by gray matter. Now, if you look at this brain picture right underneath me, you see, or you're seeing what's called a corona cut of of a brain. This is a brain that's looking straight at you. If I cut off the front of my head, this is what you would see. And you'll notice that on the outside we have gray matter. This is where the Brodmann areas are, all 52 of them. And these gray areas are actually comprised of a very special part of the neuron. This is where you find cell bodies and dendrites and terminal buttons. So these are sort of the chemical communicating parts of the brain where chemicals are released. And so you'll notice that this is your gray matter and this is where your Brodmann areas are. Now you notice that this brain is kind of wrinkly, and it turns out that if you wrinkle the brain, since only the outside surface area is gray, if you wrinkle the brain up, you actually increase the amount of gray area or surface area in the brain. They say if you took a human brain and unfolded and sort of split it open and flattened out all the gray areas and moved out all the wrinkles, that the amount of gray area would be about the same as a sheet of newspaper. That's a lot of area, and it's all wrinkled up and then stuffed into your skull. Now, each of these areas sends information to other areas, and each one of these areas receives feedback from other areas. So, your memory area sends information to your personality area sends information back to your memory area, your visual area does the same thing. So, each area has both efferents and afferents. Now, if you're looking at this brain, you'll notice that it appears to be sliced right down the middle. You see this big split in between it, and this is what's known as the central sulcus. And what it does is it divides the brain into a left hemisphere and a right hemisphere. So if you're looking at this person, they're actually looking at you right now. So what appears to be your left would be their right hemisphere, and what appears to be the right would be their left hemisphere. And you'll notice that these two hemispheres are completely separated except for this band of tissue right in the middle. You see, I have a red semicircle. This represents the only connection between these two halves of the brain. All right. And so these are hemispheres. Now, the crazy thing is, your left hemisphere controls the right part of your body, and your right hemisphere controls the left half of your body. Your left hemisphere feels the right half of your body. Your right hemisphere feels the left half of your body. Does that make sense? This is known as the contralateral design, and this appears to exist because sometime during the first couple of weeks of brain development in utero, the brain seems to twist. It goes through this little twisting pattern. And what happens is the brain twists relative to the body so that you have this opposite contralateral design. Now, within each of these hemispheres, you can further divide each hemisphere into four lobes. All right, we're going to talk about that in a little bit later. And as I suggested to you, there are individual spots within the lobes. Now, here's the crazy thing about these Brodmann areas. Not all of these Brodmann areas really have a specific activity. That is, you can see brain damage there, and a person doesn't lose a specific brain activity. That's because some of these Brodmann areas are what we call functional areas. They actually have a task, if you will. They may feel your body, they may have your personality, they may have your memory. But then there are also areas in the gray matter called association cortices, where these two areas communicate together. On Tuesday, we talked about how different Broadman areas might communicate with one another, and I gave a really weird assignment that I don't think any of the class liked. And what we talked about was different Broadman areas. And then we talked about how these two different Broadman areas might actually work together. And the way they work together is by meeting in what's known as an association cortex. All right. If you have any questions, please don't hesitate to type them in the chat bar. I'm going kind of quick and I can't look at your faces to see if you're lost or not. So if you are lost, please let me know and I'll be more than happy to go back and answer a question. Okay, now here's the deal. We have all these different spots in our brain, in our gray matter called Brodmann areas. And these areas either have specific functions or they uh, coordinate functions in association cortices. Now, here's the thing. These brain areas need to be connected, and they are connected in what's known as the white matter. Let's actually go back. Do you see all the white matter in the middle of this brain? This white matter is not goo. It is actually lots and lots of axons, and there are so many axons together that they look tangled up to you and they look like just a pile of white goo. And it's white because these parts of the neurons are covered with white fatty myelin tissue. So this white matter in the middle of this, it's not just goo, it's actually lots and lots of axons that connect these different brain areas, and it looks white because they are all covered with a white fatty tissue. Now, if you were able to image uh, those wires in your brain, those axons, they would look something like the picture below me. This is what's known as a DTI image, a diffusion tensor imaging. And what a DTI can do is it can actually view and give you an image of what the white matter connections, the axons, what their pattern of connection is. And if you look at this picture below me, this is the same picture as before. This is a coronal cut. Okay, so you're looking right at me. But now we've removed all the gray areas, and the only thing you see are the axons that connect these different parts of the gray matter. So the gray matter's gone, the white matter's gone, and all we see are the axon connections. All right. And so these axon connections tend to run in three general ways. The first are from the left half of the brain, left hemisphere of the brain to the right hemisphere of the brain. And you see those are color coded red. And what you'll see at the top, you'll see a side to side connection. That's called your corpus callosum. And then if you see at the bottom of that image, we're going to be talking about the pons in a minute. And that connection at the bottom is in the pons going left to right. Then the second thing you'll see, you can't really see it because we're looking straight ahead, but if this image turned its head sideways, you would see what we call association fibers. So you have axons going from side to side. So not only do we have commissural fibers that go from left to right, but you have connections, axons that go from front to back, connecting different parts of the same hemisphere. And then the biggest image that you'll see here are the blue fibers, and those are projection fibers that are going from your conscious parts of your brain down to your spinal cord and to your body. And you'll notice that there are a ton of these connections because your brain has to be well connected to your body. I hope that makes sense right now. Now, here's the deal. Most myelination that occurs when we're born, the reason that we are so helpless, is because these wires that you see below me don't work very well because they're not myelinated. And because that's not in your brain when you were born. However, over the first couple of years of your life, your brain is going to begin to myelinate all of these different pathways. As these pathways myelinate, you will begin to develop control over your different functional abilities. And you know what? It takes 25 years for your brain to fully myelinate. The last part of your brain that myelinates is your frontal lobe, and that's the part that creates your personality. All right. Now, the weird thing is there are disorders. where people have where their myelin sheath around their axons begins to disintegrate during their lifetime. And typically you'll see this on axons in the brain, the optic nerve, or in the spinal column. One example of this that you may have heard of is what's known as multiple sclerosis. And what happens in multiple sclerosis is your immune system begins to attack the myelin sheath in your spinal column, and it leads to an inflammation of these cells, and they wind up dying. And this damages the myelin sheath, and it scars the myelin sheath. And so people with multiple sclerosis, you notice, are going to have difficulty controlling parts of their body. All right, any questions? Two principles of the brain. Modularity, you have all these different modules in your gray matter, and all of these areas are connected. So modularity and connectivity, two very important characteristics for you to understand for the brain. Now, what I'd like to do is go through and introduce you to some of the different areas in the brain whose functions have been identified. Now, what we're going to do is we're going to start out with we're going to start out with structures that are underneath the cerebral cortex. All right. Now, typically, what you're going to find in the brain is that you can organize it from back to top, back back to front. Okay, and 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 you can uh, from back to front. And what you're going to find is that the structures that occur in the back lower part of the brain are going to control automatic basic biological functions. And as we move towards the cerebral cortex, what we're going to find is that our behaviors that are controlled are more human like and more complex. All right. So, uh, so we're first going to start by talking about subcortical brain structures. We're going to talk about the hindbrain. This is the oldest part of the brain, evolutionarily speaking. And if you look at these brain structures, you're going to notice that some of your most simple animals are going to have these same brain structures because these animals have the same functions that we do. So I like to think of this as your lizard brain, if you will, because this is the part of the brain that controls the same functions in you and a lizard. We'll start with the brain stem. It has the medulla and the pons. And if you look at this picture underneath me, you can see these areas. The medulla is going to be responsible for your breathing and your heart rate. And your pons is going to be important for controlling physiological functions when we sleep at night. These are your lower brain functions that are controlled in the brain stem. Now, for the exam, there will not be a picture. I take that back. I, I'm, I may not have a picture in the exam. Actually, I take that back. There will probably be a picture in the exam. So it might be useful to know where these brain structures are. So we're going to have the medulla and the pons. Now, if you look behind the medulla and the pons, you see that purple structure. It's not really purple in real life, but it's purple just for this illustration. You'll notice that we have the cerebellum, and this is the part of the brain that controls your balance and your motor coordination and your motor learning. If you've ever learned to play a guitar, if you've ever learned to play a sport, actually, if you can walk, and we can all walk, um, these behaviors involve lots of body parts working together in a coordinated manner. Your cerebellum is the part of the brain that learns, that changes as your body parts learn to walk, as your body parts learn to work together. So when you learn to walk, you have to learn to move your left and right foot at the same time in different directions, and they're doing different activities. But those activities need to be perfectly correlated. If you've ever seen somebody walking around that has trouble walking, or they walk with a really funny gait or odd gait, or they just seem really uncoordinated, chances are they've got cerebellum damage. Okay, so that's a very important part of your brain for coordinating complex motor behaviors. Now, one other structure I would like to point out is your substantia nigra, which is sort of in the mid part of your brain. It's in it between the hindbrain and your cerebral cortex. And the substantia nigra is part of your reward circuit. So when you do things that make you feel good, have sex, eat food, take drugs, your substantia nigra is activated. And your substantia nigra also seems to be important in initiating behavior. If you suddenly decide to get up or to lift your arm, your substantia nigra is the part of your brain that kicks everything into gear. And you know what? Your substantia nigra also appears to be the place that creates dope, is responsible for dopamine production and is associated with Parkinson's disorder. Okay, dopamine helps smooth out your motor movements, and the substantia nigra produces dopamine to help smooth out your motor movements. When the cells in the substantia nigra begin dying, dopamine isn't produced, and the behavior of the person becomes shaky, right? And that's one of the characteristics of Parkinson's disorder, these Parkinsonian tremors. And so the substantia nigra is a very important part of the midbrain. Now, the midbrain is also has your basal ganglia, but your basal ganglia isn't discussed in the book, so we're just going to pass that on and talk about that maybe a little later in the semester. Okay. Now, these other structures, there are structures that are still under your cerebral cortex that are newer, more evolved than those hindbrain structures that I told you about, and they control more complex behaviors. Now, these behaviors are still subconscious. You don't really think about these things, you don't know that they're going on, but these are other behaviors that help create the final complex behavior that is you. All right. And so we're going to talk about these subcortical forebrain structures. By saying they are forebrain, we're saying they are higher up in the brain, and we are also saying that they are evolutionarily newer than the hindbrain structures. All right. Now, these subcortical brain structures that I want you to be familiar with are the thalamus, the hippocampus, the amygdala, and the hypothalamus. All right. When we get to chapter 5 and we talk about sensation and perception, feeling, seeing, hearing, smelling, uh, and pain and heat and all of those different sensations, I'm going to suggest to you that all of these sensations, except for smell, are processed through the thalamus. This is like a sensory gateway. All of your senses come through the thalamus. And in fact, there's an area in the thalamus, not on the exam, the superior colliculus, and this is the part of the thalamus that coordinates your eyes with your hands. So I see my coffee cup over there, and my eyes are going to tell my hand exactly where to go to get myself a drink of coffee. How did my eyes coordinate with my hands? Well, that information was coordinated in the thalamus, which integrates and regulates conscious sensory and voluntary movements. We may or may not get time to talk about this in class, but dysfunction associated in the thalamus and the basal ganglion uh, may be responsible for the symptoms that go along with Tourette syndrome. In chapter 7, we're going to talk about memory, and I'm going to suggest to you that human beings have to process memory in their hippocampus in order for those memories to become permanent. So most of you probably know most of you who grew up in the US probably know who the first president of the United States was. Of fellow named George Washington. In order for you to know that information, it initially had to be processed through this hippocampus, which is responsible for the formation of memories. Now, as a side note, you want to know the hippocampus is also responsible for spatial location. So, if you really want to improve your memory, use location when you're trying to create memory cues for things. We're going to talk about that in chapter 7. I'm going to show you how to use the method of location to become an incredibly good memory person. Now, in chapter 10, when we talk about motivation and emotion, I'm going to talk to you about this structure called the amygdala. The amygdala is your primary animal emotional center. It is the part of your brain that notices objects and quickly decides whether or not you are afraid or interested in a particular object. The amygdala is the emotional center of your brain. And located right underneath the thalamus is a structure called the hypothalamus. Hypo means beneath, so does the structure below the hypothalamus. And this is the part of your brain that regulates bodily functions and motivates behavior. It's the part of your brain that tells you and we'll talk about this in chapter 10, motivation and emotion, along with the amygdala. The hypothalamus tells you you're hungry. The hypothalamus tells you you're thirsty. The hypothalamus tells you you're hot or cold or interested in sex. The hypothalamus, it tells you when it's time to go to sleep and when it's time to wake up. So the hypothalamus regulates body functions and motivates your behavior. Hopefully everybody's got those structures, right? Now, here's the deal. All the structures that I've talked about up to now are what we would call subcortical brain structures, and they all control functions that are below your level of awareness. So you're not aware that your hypothalamus is motivating your hunger or making you go to sleep. You're not aware of your medulla causing your heart to beat. However, anything that you are consciously aware of is processed through this thing called the cerebral cortex. And this is that wrinkled part of the brain that we talked about a little bit earlier. And this is the part of the brain that most people think of when they think of a brain. They don't think about the subcortical structures, they just think about the cortex. And what I'd like for you to do is to be able to tell me what the functions of each of these parts of the cerebral cortex are involved in. Okay. Now, what I'm going to do is I'm going to go through these 4 lobes of the brain and some of the specific parts in the in the brain in these cortices. Now, here's the deal. On the exam, I reserve the right to do what I like to call brain damage. And so what I'll do is I'll say, Timmy has lost the ability to hear anything. Which part of his temporal lobe was damaged? And you'll say it's the auditory cortex. Or I'll tell you that Mary um, has trouble understanding where she is in space, and she also gets lost and can't find her car. Which part of her brain, which of her lobes is damaged? And you'll say, oh, that's clearly the parietal lobe. All right. Or I might tell you, Paul Broca's patient Tan couldn't say any words except for Tan. What part of his frontal lobe was damaged? And you'll say, oh, it's Broca's area. All right. And so that's how we're going to play this on the exam. And I want each and every one of you to know the function of these different areas. Most of these different areas function have been learned through lesioning studies, through case studies uh, that have demonstrated the link between these areas and specific brain functions. All right. Now, each side of your brain, each hemisphere is divided into 4 lobes. You have a frontal lobe, you have an in the front, you have an occipital lobe in the back, you have a temporal lobe on the bottom, and you have a parietal lobe on the side towards the back. These are 4 lobes, and they're the reason we call them lobes is because you can actually take that hemisphere and pull it, that left or right hemisphere, and pull it apart, and you can see that these are 4 functionally different parts of the hemisphere. And you'll see them in color here. You'll see the frontal lobe is the largest of the lobes. It's the largest in humans. And so it's going to contain our most human-like behaviors. You'll notice right behind that is the parietal lobe. And then at the very back in yellow is the occipital lobe. And then the green temporal lobe is sort of at the bottom, going from back to front of the hemisphere. Now, if you look within each of these lobes, I can then, to the right of that picture, you'll notice that you have your frontal lobe in pink, and at the back of your frontal lobe is this thing called your primary motor cortex. And the frontal lobe and the parietal lobe are divided right here. And so you'll notice that there's a somatosensory cortex right behind the primary motor cortex. That primary sensory cortex is in the front of the parietal lobe. All right. Now, looking at the temporal lobe, I want you to notice that it has a specific area just for hearing, the primary auditory cortex. And then as I started the lecture off, you'll notice right there at the back and the underneath part of the uh, temporal lobe, you'll notice that there's the fusiform face area that I told you about earlier. Okay. And then in the very, very back tip of the occipital lobe, what you're going to see is the primary visual cortex. All right, now let's go over and talk about these really quickly. All right, in the frontal lobe, the prefrontal cortex. Now, this picture doesn't actually have, you know what, is it the next picture? No, it isn't. It's one in your book that I haven't shown. Your frontal lobe's actually got three areas I'm interested in you being aware of. The actual front part of your brain is called your prefrontal cortex. And this is the part of the brain that makes you uniquely human. This is the part of your brain that allows you to pay attention, to control your impulses. And this is the part of your brain that's responsible for your personality. If you'll look in your text, you can read the story of Phineas Gage, the railroad worker, and there was an explosion, and he damaged the frontal part, this prefrontal cortex, and it caused his personality to change, and he lost his ability to control his impulses. So I want you to know the prefrontal cortex. And then I also want you to know that in the bottom part of your prefrontal cortex, right in front of the motor cortex, there's an area called Broca's area. Right here, it's on the left side of your brain only. Here's the crazy thing. Speech seems to be processed for the most part in the left side of the brain. So Broca's area is in the left side on your frontal lobe. And that's the part of your brain that allows you to form words. If you have damage to that, you will have difficulty speaking. You may be able to say more than one word, but you will have great difficulty getting those words out of your mouth. And then we're going to talk about this in a couple of minutes, but then you have this thing at the back of your frontal lobe called the motor cortex. And this controls the voluntary movement of your body. If you lift your left arm up, your right motor cortex just activated it. If you lift your right leg up, like I just did, my left motor cortex controlled that behavior. So your motor cortex controls the movement, initiates the movement of your body. Now, the movement of these different parts has to be integrated in the cerebellum, which allows them to work together, right? But the frontal lobe, the motor cortex in the frontal lobe is the part that actually activates that particular body part when it's time to move it. So when I blink my left eye, my motor cortex on the right side is causing that to happen. The same thing when I wiggle my nose. My motor cortex is causing my nose to wiggle. So those are the three areas I want you to know in the frontal lobe. Now, right behind the frontal lobe and on the bottom there is the temporal lobe. Basically, what I want you to know is that there's the primary cortex where we register pitch, loudness and duration of sounds is located right there in the temporal lobe at the very top. And then I want you to know that the fusiform gyrus, which is your facial recognition part of your brain, is also located in the temporal lobe. And then I want you to know that in general, if you think about it, your knowledge for people's faces is a memory. I want you to know that memory in general is stored in your temporal lobe. If you have damage to your temporal lobe, you will lose your ability. You may lose your ability to remember things that happened to you earlier in life. Now, I'm going to tell you a crazy story in chapter 7 when we get to cognitive psychology, and I'm going to tell you how the temporal lobe and the hippocampus work together to create memory. So your hippocampus makes new memories, and your temporal lobe is where old memories are stored. So if you have damage to your hippocampus, you won't be able to make new memories, but you'll be able to remember the things you used to know. On the other hand, if you have damage to your temporal lobe, you will lose the memories that were stored in that area of your temporal lobe, but your hippocampus will be able to make new memories in other parts of your brain. Kind of crazy, huh? All right, so I want you to know those structures. Okay, in general, your parietal lobe is responsible for the conscious feeling of your body and its spatial orientation. So, right now, I want each and every one of you to point, as you're sitting here watching this lecture, point in the direction in which your car is parked. Mine is parked right over that way. I don't have to see it, I just know where I am relative to my car based on where I'm sitting in my house. So I can tell you that my car is outside that direction, even though I can't see it. I'll bet each and every one of you can tell me right now which direction you would have to walk in order to get to your car. In fact, I'll bet each and every one of you can navigate the pathway through the doors, taking lefts and right turns to get to your car. Your ability to orient yourself in space and to move about in space is controlled by your parietal lobe. All right? On the other hand, your sensory cortex, that strip in the front of your brain, right behind your primary motor cortex, is the part of your brain that consciously feels touch. So if I stroke my right cheek right now, my sensory cortex on the left side feels that that stroking. If I twist my left finger and it hurts right now, my finger spot on the somatosensory cortex tells me that my finger hurts. All right. So your somatosensory cortex feels the parts of your body. So if you think about it, you've got these two strips in your brain, one that moves your body parts and one that feels your body parts. Now, in your occipital lobe, you have what's known as a primary visual cortex, which builds images. Remember, I told you earlier that there's a bunch of different steps to building a visual image? Well, your primary visual cortex sees lines, it sees movements, it sees color, okay? And then, and you'll notice that that's that yellow dot right at the back of the occipital lobe. And then right outside the primary visual cortex, you have what's known as a visual association cortex. And this is where these lines, this shape, and this color, and this movement all come together to create images. Now, once you create these images, you're then going to send them to your temporal lobe so you can try to remember what these images actually are. Broca's area is not a cortex, it is inside a cortex. Okay, so we have four cortices. That's a great question, Cole. So we have the uh, right. It's actually it would be called a goodness me, I forget what it would be called the functional area would probably be the best way to describe it, Cole. All right, finally, before we go, I've got a couple of minutes. I want to talk specifically about the motor cortex and the sensory cortex. If you take a look at the picture I have over here on the left, you see sort of what appear to be slices taken out of the motor cortex and the sensory cortex. So imagine that what I've done is I've taken that light blue thing, I've cut a slice and pulled it out so that you can see it with the coronal view, just like we started this class. Okay. And I've done that for the motor cortex and the sensory cortex. And then what you see on here are the parts of the body. And what they're trying to represent in this picture is that the different parts of your body are mapped onto the gray matter in these two strips of cortex. All right. So you'll notice that there's space that feels and moves your tongue. There's space that feels and moves your teeth. There's space that feels and moves your gums, your lips, your face, your nose, your eye, your brow, your neck, your thumb, your fingers, your hand, your arm, your shoulder, your neck, your trunk, your hip, and all the way around. And so each part of your cerebral cortex. Now this comes at birth, and this is just how your brain is organized. So you have a face area in your somatosensory cortex that feels things that touch your face. You have a face area in your motor cortex that allows you to move different parts of your face voluntarily. Okay. Now, what you should notice in looking at these pictures is that your arm is very small on this map and your face is very big on this map. This demonstrates a principle of the motor and somatosensory cortex with regard to our body. The size of the area that is given to a body part in your brain is not dependent upon the size of the body part, but how complex its behavior. And so you have way more space on your brain to move your lips than you do to move your entire leg. Why? Because lips have to do lots of things. They have to talk, they have to eat, they have to kiss. Your lips have all these very, very complex movements, whereas your leg just goes up and down, and maybe a little side to side, right? So the size of the functional area represented in the sensory and motor cortex is dependent upon the complexity of the body part and not necessarily the size. Now, again, I want to remind you of this thing called the contralateral design. The left hemisphere feels and moves the right side of your body, and the right hemisphere feels and moves the left side of your body. Check this out. I'm really going to trip you out in 2 weeks when we talk, or 3 weeks when we talk about sensation and perception. You can draw a line down the middle of your field of vision, and the left hemisphere sees the right side of your visual field, and the right hemisphere actually sees the left half of your visual field. Now, the other thing is I want you to know is that if you damage part of your motor or sensory cortex, another part of your motor cortex will take over controlling the body part that was originally supposed to be controlled by that motor or sensory cortex. On the other hand, if you lose a body part, that part of the brain that used to control that body part will find something else to do. So your brain can actually change its design in response to damage. This is known as plasticity. Plasticity. When you get to chapter 5, there is a video of Ben Underwood, the blind boy who learns to see with his ears. This fellow lost his eyes to cancer when he was 3 years old, and his ears learned to take in sonar information and build a visual picture in his mind. So plasticity is also a key component of the nervous system. And then finally, I want to tell you that my father lost his leg to cancer about 20 years ago, and he still feels that missing leg. This is called phantom limb pain. Just because you lose a body part doesn't mean that that brain area isn't there looking for feedback from that body part. And there are some people, most people actually, who lose a body part will actually feel this phantom limb pain. And there's a cool treatment. If you remind me about it, maybe this week or next week, I'll show you. It's called the mirror box treatment. where you can use the eyes to actually fool a person into making a body part a missing body part quit hurting. Okay, that's all I had to say today. So we are finishing up the introduction to biopsychology. Yes, it was a brief introduction and I just hit the high points. But what we're going to do over the course of the semester as we talk about these different areas of psychology is I'm going to go back and visit this biopsych chapter again and again. And we'll talk about the behavioral ability or what's going on. So next week we're going to talk about sleeping and I'm going to talk about the brain, the parts of your brain, the hypothalamus, the pons, and the thalamus, and how they function during the process of sleep. Does anyone have any questions about the lecture right now? Don't forget, we have our optional webinar tonight at 7 p.m. Tonight is the early webinar. It's at 7 p.m. And you can either do the discussion board or you can come to the webinar. If you've already done the webinar on Tuesday, you do not need to come to the webinar tonight to get credit. All right, we are at 9:55. I am right on time. Holy cow, I feel good today. Um, I don't see any questions in the chat bar, so I'm going to go ahead and log off this broadcast. Remember, if you do have a question, you can send me an email or a text, and I would be more than happy to answer it. Have a great day, and I will see you tonight in webinar. Take care.